Hi, everyone. Welcome to the Australian Biocommons AI in the Life Sciences webinar series. My name is Georgia. I'm the Australian Biocommons BioCloud Training and Communication Officer, and I will be your host for today. This webinar series is all about how artificial intelligence is being used in life sciences right now in real-world research. You'll hear directly from experts working across biology, medicine, agriculture, and environment as they share how AI is helping them and their research. Before we begin, we'd like to take a moment to acknowledge the traditional owners and their custodianship of the lands on which we meet today. In my case, this is the Gadigal people. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connection to country. We recognize their valuable contributions to Australian and global society. Today, we're thrilled to welcome Dr. Maisie Li and Dr. Túlio de Lima Campos. We'll hear from both of them, and we'll have time for questions at the end of their presentations. We'll start with Túlio. Túlio is a multidisciplinary researcher with extensive expertise in AI, bioinformatics, and genomics. He holds a PhD in veterinary sciences from the University of Melbourne, where he focused on assessing machine learning approaches for predicting essential genes in model organisms and parasites of veterinary importance. Currently a public health technologist at the Osvaldo Cruz Foundation in Brazil and an honorary fellow at the University of Melbourne, Túlio actively contributes to genomic studies of pathogens and parasites. Welcome to the webinar, Túlio, and I'll now hand it over to you to start the presentation.
Thanks for the kind introduction, Georgia. It's a pleasure to be here today to speak about AI, and particularly considering the relevance of the topic that how it has gained traction in recent years. So today I'll be talking about an important and quite common application in machine learning and in AI, and that is binary classification. Several concepts to be discussed here are not necessarily new, and but still require considerations during development and evaluation of the different approaches. So from data to algorithms and the appropriate interpretation of results. So the field of artificial intelligence, binary classification, is one of the most fundamental problems that we tackle. And it involves teaching a model to distinguish between two categories. For example, whether a tumor is malignant or benign, or whether a gene is a biomarker for a disease or not. And these tasks fall under the umbrella of supervised learning, where we train algorithm on labeled data. By learning patterns in this training data, the model becomes capable of predicting the correct class for new unseen examples. So binary classification is not only essential in decision-making in biology and healthcare, but it also serves as a foundation for more complex classification systems. And the quality of the data and how we represent the process strongly influence the success of these approaches, making classification, binary classification, very important in the use of AI. So during my PhD, I had the opportunity to work on a challenging project focused on prediction of essential genes in eukaryotes. And that is genes that are essential for survival or reproduction in organisms. And this work was conducted under the supervision of Associate Professor Neil Young and co-supervision from Professor Robin Gasser and Basil Corhonen at the University of Melbourne. And our main goal was to model the complex biological question as this biological question is a binary classification. So given the large-scale molecular data sets available for model organisms, could we then predict essential genes based on these data? And this approach allowed us to identify and to apply machine learning methods in a problem that is linked to functional genomics. And these offered insights into the biological determinants of gene essentiality. And this provided a basis for predictions in no-model species for which experimental validation is difficult, such as parasitic helminths. And by carefully designing features and selecting appropriate algorithms, and interpreting model outputs in the biological context, we develop models with strong predictive performance and biological relevance. So in the next slide, I'll go through the steps, challenges, and some insights from this journey, highlighting not only the technical side, but also the importance of biological interpretation and domain expertise in making AI meaningful in the life science. So before we even think about training machine learning or AI classifier, it's crucial to deeply understand the problem we are trying to solve, the data we have or we plan to generate, and consider any potential biases hidden within this data. A well-defined problem guides how we collect or label our data, what features we consider, and how we evaluate success. And the quality, diversity, and completeness of the data are just as important as the algorithm itself. And as you know, garbage in, garbage out. If our data set is unrepresentative or reflects historical or experimental biases, the model will inevitably replicate these flaws. And this can lead to poor performance, misleading results, or even harmful decisions. So successful classification in AI isn't just about the models. It's about thoughtful, ethical, and informed data, as well as problem design from the very beginning. In the biosciences, transforming raw biological data into meaningful features is a critical step for building reliable classification models. So feature selection starts with choosing a wide range of measurements, whether it's gene expression levels, clinical markers, or environmental variables, and ensuring they are accurate, consistent, and well documented. And then comes feature engineering, where we translate this raw data into biological relevant indicators or characteristics linked to these classes. And finally, feature selection helps to filter out noise and focus on the variables that truly contribute to the biological question at hand. And this is specifically important when dealing with high-dimensional data sets, where there are too many irrelevant features that can make can disturb the models. Along features and so strong features aren't just mathematical, and they reflect biological mechanisms. And getting them right away and turning into data to organize these features for discovery. And feature selection techniques, they help identify the most relevant features for the model. For example, recursive feature elimination that iteratively removes the least important features based on the model performance, and therefore keeping only the relevant features. And another common feature selection method is lasso that penalize features that are less informative and shrink their coefficient towards zero during the model training. So when training a classification model, it's essential to evaluate the performance on data that hasn't seen before, the model hasn't seen before. And this is where data splitting comes in. We typically divide our data set into at least two parts: the training set used to fit the model, and the test set used to assess how well it generalizes on new data. Sometimes we also use a validation set to fine-tune the parameters before touching the test set. But with limited data, setting aside data set just for this purpose can feel like a waste. And that's why cross-validation becomes very powerful. In k-fold cross-validation, we divide the data into k equal parts, and we train the model k times, each time using a different fold, and the test set with the remaining folds for training. And this way, data points, so we have the data that's used for training and testing, but never at the same time. So cross-validation provides a more reliable estimate of our models' performance and helps prevent overfitting, specifically in high-dimensional or small sample scenarios, which are common in the biosciences. Ultimately, smart data splitting and cross-validation ensure that our models are not just good for memorizing, but are capable of generalizing. So when tackling a classification problem, it's really a one-size-fits-all problem. So different algorithms make different assumptions about the data, about its distribution, feature relationships, and the underlying patterns. For example, logistics regression performs well when features are linearly separable and relatively independent. But it might struggle when there is collinearity or complex nonlinear relationships. And by contrast, tree-based methods like random forests or gradient boosting can handle heterogeneity and correlated variables much better, though they come with their own trade-offs in interpretability and overfitting risk. That's why it's so important to try multiple algorithms and tune their parameters. The structure of your data and the way how balanced the classes are, whether the features follow a normal distribution, can greatly influence what works best. And take, for example, expression data. And if you use a support vector machine, it might work well when classes are clearly separated in high dimensional space, while a random forest might outperform when dealing with noisy, redundant features. So by exploring and comparing models systematically, we can identify the approach that best captures the signal in the data, not just what's on the surface. So choosing the right performance metrics is critical when evaluating the classification model. So accuracy might seem the obvious choice, but it can be misleading, specifically in imbalanced data sets, which are common in biosciences. That's why we need to look beyond accuracy and consider metrics like precision, recall, and the F1 score. For example, precision tells us how many of the positive predictions were correct, while recall tells us how many actual positives were captured. And depending on the context, we may want to maximize recall. For instance, in cancer screening, where missing a true case could have serious consequences, or precision in scenarios where false positives can lead to costly invasive follow-ups. And there are other metrics like area under the ROC curve, area under the precision recall curve, that are also useful metrics for summarizing performance across different thresholds. But ultimately, defining the right metric means it will be really problem dependent, and we need to consider the real world scenario to define the best metrics. And without that alignment, even the technically strong model might fail to deliver meaningful or suffer outcomes. And speaking of class imbalance, this is a very common problem, specifically in biosciences, where the positive cases, for example, patients with a rare disease, can be vastly outnumbered by negative cases. And when one class dominates, a model can become biased toward predicting the majority class, achieving high accuracy while completely failing to identify the minority classes. And this only reduces the usefulness of the model, but can lead to critical errors in real-world scenarios. And to address these, we can try resampling techniques like oversampling the minority class or undersampling the majority class to balance the data set. And alternatively, we can use an algorithm level solution that adjusts class weights to the model and penalize misclassifications in the minority class more heavily. And some algorithms like extreme gradient boosting and regression models have this support. However, artificially fixing class imbalance has trade-offs. For example, oversampling may lead to overfitting, specifically or especially when synthetic examples are too similar to real ones. And undersampling might discard valuable information, and change class weights can make training unstable if not tuned correctly. And that's why it's essential not only to acknowledge the class imbalance of the data, in that case, if that's the case, but to evaluate your model using sensitive metrics like precision recall and F1 score. And in the end, addressing class imbalance thoughtfully ensures that the model isn't just accurate, but also fair and useful. And when building classification models, two common pitfalls to watch for are overfitting and underfitting. Overfitting happens when the model learns on the training data too well, including the noise and random fluctuations. And then as a result, it performs excellently on the training set, but poorly on the new unseen data because it hasn't captured the underlying patterns. And this is this is like memorizing answers rather than understanding the concepts. On the other hand, underfitting occurs when the model is too simple to grasp the complexity of the data, and it fails to capture the important relationships and show poor performance both on training and test sets. Think of that as not studying enough to solve a very basic problem. So balancing these two is critical. And then we have techniques such as the cross-validation, regularization, and pruning to help avoid overfitting or controlling model completely, while feature selection and selecting more expressive algorithms can help prevent underfitting. And the goal is to build a model that is capable of generalization, meaning it performs reliably on new data sets, which is essential in real-world applications, specifically in fields like biosciences and healthcare. So building an accurate classification model is only part of the challenge. Interpreting the model decisions is equally important, especially in sensitive fields. Model interpretation helps understand why the model makes certain predictions, which builds trust and adds troubleshooting and can even reveal new scientific insights. And there are several methods to interpret models. For simple models like logistic regression, we can look directly to the feature coefficients to see which variables are predictors. And for more complex methods, like random forests or neural networks, there are techniques like shape values or Lime that provide focal explanations by estimating the contribution of each feature to individual predictions. And another approach is feature importance ranking, which helps identify which features the model relies most across the data set. So interpretive models also help identify biases or errors in data and ensure decisions align with the domain knowledge. And ultimately, model interpretation bridges the gap between black box AI and actionable understanding, empowering the researchers and clinicians to make more informed decisions with confidence. So interpreting model predictions goes beyond the numbers, and it requires careful biological interpretation. A model might highlight certain genes or biomarkers that are important, but without understanding the biological context, these findings can be misleading or meaningless. And biological interpretation ensures that the model's predictions align with known science and plausible mechanisms, helping to validate results and generate new hypotheses. And this connection between model interpretation in biology is crucial. When we use tools like SHAP or feature importance, we get clues about what drives the model's decisions. But then translating those clues into meaningful biology means collaborating with domain experts who can assess whether the signals reflect the pathways, the disease mechanisms, or the experimental artifacts. So careful consideration makes AI not just a predictive tool, but a powerful partner for discovery and decision-making, ensuring that computational insights for meaningful advances in health and sciences. So in summary, building effective classification models in biosciences requires more than just powerful algorithms. It starts with deep understanding of the problem, the data, the challenges in the class imbalance, overfitting, underfitting, carefully crafting and selecting features, splitting data thoughtfully, and trying a multitude of algorithms, and helps ensure robust performance. Equally important is defining the right metrics and interpreting the models responsibly, linking computational results back to meaningful biological insights. And by integrating these steps with domain experts, we can turn complex data into trustworthy and actionable knowledge, advancing both science and translational applications with confidence. So thanks for your attention, and over to you, Georgia. Thank you.
Thank you, Túlio. So Maisie is a postdoctoral fellow in the Breeding and Innovations Group within the Agriculture and Food Research Unit at CSIRO. Her current research focuses on designing the collaborative intelligence platform with applications in genomics and bioinformatics. Prior to this, Maisie was a research associate at the University of South Australia, where she developed drug repurposing strategies for COVID treatment. She earned her PhD in bioinformatics from University of South Australia, where she specialized in computational methods for breast cancer prognosis. Maisie's broader interests lie in the development and application of data mining, machine learning, and causal inference techniques to address real-world challenges in life sciences. Welcome to the webinar, Maisie.
Yeah, thanks, Georgia, for the nice introduction for me. And so I'm Maisie Li, and I'm currently working at the CSIO. And today my presentation is about towards human AI collaboration in genomics and bioinformatics. And thanks for the opportunity to share my AI journey. So this title is more about what I'm currently doing and hopefully continue in the future. Before dive into my current work, I would like to share with you my AI journey for life sciences. So my background is computer science. My first project related to life sciences is about the protein function prediction. So here is a pipeline that I use to predict the protein function. So I maybe introduce this a bit. So in this project, there was so we start from the protein sequence data in the fast format, and then the very important part is we need to convert this sequence data to machine understandable or algorithm processable data. So we are using the feature extraction method to convert this sequence data. I just want to okay, what? No, so just want to highlight something. I don't. Okay, so so we are convert the sequence data, and for the training part, we need labels which indicate the function of the protein. So with the both data, we transfer them to a numeric matrix, so which the rows indicate the protein and the columns is the features and the labels. All of them, so this matrix and numeric matrix. So for this, in most of the cases, we don't have much protein, so we have more features. So in this case, we're called a small sample problem, so where it's not good for training AI models based on such data could have a high dimensional problem. So as Túlio mentioned, so we have a technical called a feature selection method, so which will select the most relevant or important features from this original feature space. And then we based on the selected features, and then we use it to training classify for these problems. So at that time, I mostly use SVM, so which is have strong foundation support and also have a promising performance. When we're using this model, but however, in the training phase, we need tuning hyperparameters to get the optimized model. For example, a simple one is a SVM model with a linear color. So there was one parameters is a C, so which is control the trade-off between the maximizing the margin. So here is and the minimizing the classification errors on the training data. So when we fine-tune the hyperparameters, we need to understand what these hyperparameters associated with. So that's also, for example, if you're having so if you have a very in this case, if we have a smaller C, so we get probably get a big classification errors on the data based on the data. And so the key message here is we really understand what the hyperparameters meaning, and usually we need training multiple hyperparameters, so you need to use the HPC to speed up this process. Yes, I'll come to this next slides. So after the training phase, so we are going to do the test phase. So we are using the same feature selection extraction method to convert the protein sequence data to the numeric matrix feature matrix this time, because we don't have a label anymore. And then we are using the same feature selection method, such as the same set of the features. And then we are fit this feature matrix to the trained SVM classifier, use this classifier to predict the labels. So that's the whole pipeline to do the protein function prediction. So this kind of method is work well on the single label problem. So I'll give an example here. For example, in the image classification task, so an image like this is only assigned with one label, which is dog. However, in the real world problems, so we usually need to assign multiple labels to a single sample simultaneously. For example, in the protein function prediction, the protein could have multiple functions. So in this case, to address this problem, we need to develop the multi-label classification method. Although a simple solution could be we're training one binary classifier for each of the label. For example, we can each of the classify just answer the question like DNA, whether this protein has DNA binding function or not. So for each label, we're training one binary, a single label classify. However, you can imagine if we have a lot of labels, we need to train multiple binary classify for that. Yeah, so and also the last problem for that is when we try training the binary classify, we are not consider the the relationship between the labels. For example, in the G ontology annotation, the labels in the Go structure are actually interrelated. So to solve this problem, I mentioned earlier, we are need to use a multi-label classification method. So yeah, I started this project 10 years before. So now they have many algorithm comes out. So this paper has nicely summarized many methods published in this domain. Yeah, after my first project, I continually apply AI models to various of tasks in life sciences. For example, I have worked on RNA tertiary structure prediction. Use the similar pipeline I had presented before. Yeah, and then in my PhD, I work on the breast cancer subtyping problem. So there I using unsupervised learning method to group the patients in distinct cluster. So each of the cluster may have a distinct clinical outcomes. Yeah, I also use a supervised learning to predict the patient survivor risk score. Yeah, so both of them are using the gene expression data. So we know that in the like cancer research, we are usually don't have too many samples, too many patients. And so then is the dimension is really high because we have more genes as a feature. So I using deep learning to learn the latent, compact latent representation to reduce the dimension of the features. And also in cancer research, the data is observational data. So that may have some selection bias. So to address, to mitigate these problems, I using the causal-based method to regulate the training process and improve the robustness of the models. And in parallel, I also work on the identification of preeclampsia biomarker, integrate biological network, and using the network inference method to identify the preeclampsia related biomarkers. Yeah, based on my those experience as similar to Túlio and last speaker and mentioned before. So when we apply the AI to life sciences problem, so we really need to deeply understand what is the research problem and carefully curate the data. And then we need to think about select the most appropriate models. Yeah, and even developer pipelines. So these models could be include the statistical AI or machine learning models. For example, a challenge of the prediction of the spatial cell position in the Drosophila embryo using the single cell transcriptomic data. We have developed multiple pipelines, including the pre-process method, feature selection, and the prediction method. And so then we find out one of the combination performs really well. Yeah, so Now, yes, in my research, I'm more focused about the genome annotation. Yeah, so I think Dr. Ben and Goody already given a very nice introduction about the AI concept and the terminology. So he classified the AI to different type of methods. So in my research, I also group the AI or tools in my research to these 4 types. Yeah, based on the timeline. So for example, there were heuristic statistical method used and in my work, and also integrated ensemble method for genome annotation, and also machine learning, deep learning method. And now we are talking a lot about the foundation models like DNA language models, ChatGPTs, and then so on, and also the AI agent. There was a lot of AI agent models. These models, I think these models they're still preactively in different tasks in genome annotation. In real practice, they are complementary. They both have each of them have strengths and limitations. If we put them together, that's really help us to solve our task. I will demonstrate this in my current work. So in genome annotation, from bioinformatics point of view, we think the assembly is really good now. However, the genome annotation is become the main bottleneck. Here we show that we only have a handful model species have well annotation, and although these model species do not guarantee have a solid annotation. For instance, then for Arabidopsis, we are have a lot of genes does not have the label indicate what the biological function. And also it's not only missing labels, the public databases full with errors. Most of the errors come from the automatic pipeline without experimental verification. So when we get the genome annotation, gene annotations from automatic pipeline, we will often find that many of the genes does not characterize in terms of their functions. So then the researchers usually need to manually curate the information from disparate data sources and get the information about the potential functions and generate the hypothesis. This manual curation process is both time-consuming and laborious. So Gene Whisperer is current the tool we developed is to streamline this process. So it is built upon AI agent, so which is the user can put query to the AI agent. So then an AI agent can use a tool to help us solve the problem. So we don't need to worry about, for example, navigating hundreds of web tabs. So it will be make the information curation more efficient. The Gene Whisperer now is incorporated in the CSIO science digital platform. We have a brief introduction and living demo online publicly. So Gene Whisperer is one of the tool. It's also AI agent as a tool. And in the whole platform is also, yeah, they have many AI agents. They have several of them are related to life sciences. So yeah, we are also interested to collaborate with external researchers to giving us feedback and which can help us improve and extend this system. This system is currently accessible within Cyro, but if you're interested, please reach us and we can discuss. So lessons from this project, we learned that AI agents, including large language models, are powerful, but they're not perfect. So the LMs and the AI agents can automate many automation tasks, but the hallucination struggle with ambiguous data or missing the biological insights. We think the expert knowledge is still essential because the complex genome mutation require carefully interpretation of uncertainty or involve biological evidence that AI alone cannot fully resolve. There were many researchers demonstrate that involving domain expert to review, guidance, and correct AI outputs can ensure their high reliability and scientific validity. In our recent preprint, we are proposed to design a human AI collaborative workflow that to integrate AI capabilities with the human expertise, enable efficient, trustworthy, and scalable genome annotation. So in this work, we firstly conduct literature review from the genome annotation, manipulation, and human AI collaboration. And we propose the conceptual framework for human AI collaborative genome annotation, which is lambda hackogram for short. And then we outline the mechanism of how effectively human AI collaboration. So here I highlight the trust is one of the key factors we would like to explore. So to understand how to build up user trust in genome annotation with the AI system, such as Gene Whisperer. So we collaborate with behavioral scientists to help us identify what the trust factors influence the users use the AI system for their genome annotation task. So they conducted a comprehensive semi-structured interview, and then they extracted the attributes that relate to the trust, and then we are together refine which trust are important to the users, and also it can apply in our system. So then the potential end users are asking the questions through the survey, show them a different combination of the attributes and at a various level, and we ask them which of the combination they would trust more. In this study, so we found that the transparency is have the most important attributes to the trust in Gene Whisperer, the tool we are developed, and then followed by the confidence and control and accuracy. Then I will also show that the relative importance of the different level of the attributes for the user's trust in Gene Whisperer. So that we found that the fully transparency of using of these tools are really important to build a trusted, reliable agent to work with users. So this kind of study really helpful to help us improve our system to make sure it's aligned with the user's expectation and needs. Yeah, so our system still keep improve, and we are very interested to know what the different user studies, user cases the user may interested, and and getting their feedback. We think is really important to evaluate our system and build a trustful AI tools for the research community. I'm deeply thankful for our colleagues for their support, and also thank for the CNTEL, FSP, and the Science Digital Program for the funding and the support. Please feel free to contact if you have any question or just want to chat. That is all from me. I'm happy to take any questions. And I'm over to Georgia.
Thank you, Maisie. Thanks to both of you. We have now time for questions. If there are many annotations in public databases already, wouldn't using a LLM trained on the current database to annotate new genes potentially magnify the issue?
Yeah, thank you, Georgia. Yeah, so the question is use LM to training the already annotated genes publicly available. So we have, yeah, so currently they'll have a lot of work on this. They're using the DNA or protein language models trained on the protein sequence or DNA or protein sequence to predict their functions. Yeah, there were many tools on this. But it's still challenging to, there are still challenges to predict the novel genes, especially the long model species. So yeah, it still need to improve. Yeah.
Another question for you, Maisie. Can you please explain which encoding method did you use to transform protein sequences to numerical features?
Yeah, thank you for the question. So in my AI agent, in my tool, Gene Whisperer, so we are not convert the DNA or RNA sequence to the features. We are using the the general AI agent, general LMs, for example, ChatGPT or OpenAI API to build up. Our process is input is human language, and your query is human language, and the AI agent is help you to use a tool to solve the problem. So that's our tool. But for this question, to how to convert the DNA RNA to the features, I think there were lots of work on this. There are transformer models as well. Yeah. Thank you for the question.
And Maisie, you talked about how the human AI collaborative gene annotation focuses on collaborative human AI systems. So can you share how this approach changes the way researchers interact with complex genomics data compared to more traditional tools?
Yeah, thank you, Georgia. That's a really great question. So this area is emerging new areas. So so we bring this concept out because now the generative AI models, ChatGPT and all these foundation models comes out, and they think the AI will replace humans' work. However, we think it's not that case. So currently, there was many works that the AI agent cannot achievable. So we think we need to bring human insight and AI or machine speed together to achieve more trustful and efficient workflow. So based on the LM agent, so don't change the traditional tool use. So we don't use a static tool developer, like we build a tool, and then we run the tool as an app. Now we're communicate with the tool, so we're just using neural language to communicate with the tool. So that's make this processes more easier for users. And also there was studies to study how these kind of tools maybe can reduce some workload from some of coding. The coding, you need the coding, so reduce this head part from the users. Yeah, so and also the AI agent can help with process data and choose AI models and help evaluation, and also write a report for users so they can streamline all the process more easily. So which is yes, very different from the traditional tool use. Yeah, thank you.
Thank you, Maisie. Yeah, you both talked about how important it is to have this collaboration between humans and AI. And Túlio, you were talking about how there are many biases that are hidden between the data that we have. So how might the way we collect and label data unintentionally introduce bias into a classification system?
Thanks for the question, Georgia. So yeah, if we need to build these models, to these machine learning models, we need to either get data that's already out there, or we need to generate the data with the different with the labels, right? So if you label incorrectly, then that's one problem that can affect the results. But also, if we have, for example, many samples with the same characteristic, but that is not representative of the total population, but the total distribution of the data. So, like people talk about, for example, like the ethnic biases in human genomics, for example, and others. So you need to acknowledge that sometimes you have these data sets, you work on them, you can classify them, and you have some metrics that you extract from there, and it's efficient for that data. To make it generalizable, we need to ensure that the data is representative of the total population. I gave one example in terms of human genomics, but it can be other examples. Like we need to ensure that there is representation in the data to build robust models. And this has implications like, okay, when we have a small data set or some data set to work on and do for a publication for science, that's one thing. The other thing is how do we bring these models to practice? And so these considerations need to be really carefully addressed.
And Túlio, you work in healthcare. So in high stakes domains like healthcare, should we prioritize model interpretability over performance? What are the trade-offs?
Yeah, that's a very good question. Thanks so much. So yeah, because the thing, especially in binary classification, we see these boosting methods like gradient boosting, cat boosting, etc., and some other approaches that, or even deep learning, for example, that can be black box models, which means like they make a decision and you don't really know like how they made that decision. So if we bring that to practice, to clinical practice, how do we whose is the responsibility if the models make a mistake when it's a black box model? But on the other hand, we may have some more interpretable approaches, which means like we can understand how the model came to that reasoning, and this can be used for more like informed decisions. Like that's AI assisted decision rather than relying on the AI for that decision. So that's a good discussion in terms of having high performance black box model or having an interpretable model that's not necessarily super accurate, but it provides some information that a doctor or someone who is making the decision can take that information and make a more informed decision. So that it can be a combination, right? You can have a black box and another more interpretable, and then whoever makes the decision, take this information into consideration. But that's a very good discussion. Thanks so much, Georgia.
Thank you. We have to leave it here for today, but I'd like to once again express our thanks to Túlio and Maisie for taking this time to talk to us. You can find out more about the upcoming webinars, workshops, and other events on our website, and you can watch the recording of this and previous events on our YouTube channel. And finally, we'd like to acknowledge our funding. The Australian Biocommons is enabled by NCRIS via Bioplatform Australia. Thanks so much for watching, and we hope you enjoyed today's presentation. We look forward to seeing you again soon. Until next time, goodbye for now.